Hello, good afternoon. You are still awake. My name is Petter, or Peter for some of you, Björkqvist from the company Verigraft in Sweden. I'm here to tell you a story about my company. And my company is a small biotech, and as you can see, we have a huge ambition. My story actually started yesterday when a journalist asked me a couple of questions. So he asked me, how old is your company? And I told him that we started this company 10 years ago. And then he asked me, what is the best moment over these 10 years? And actually my next slide, as you can see, that is about the best moment in these 10 years. That was the day when we were treating the first patient ever in the world with a personalized tissue-engineered transplant. So my company is a small biotech. We are private. We are owned by investors from Europe, from Asia, and from the US. This is what all is about. We try to provide personalized tissues for transplantation. So, we are here to talk about regenerative medicine. You will hear a lot about genome engineering, a lot about cells and cell therapies, but you will not hear that much about organs. So I will tell you a few things from my perspective. So my first slide here is a lobster, as you can see. And why a lobster? I used to have a lobster because that is the perfect introduction about what we are doing. As you may know, and as you can see here, this lobster has one big claw and one small claw. And if you break off a claw from a lobster or a crab or another animal of that type, you know that there will be a new claw growing out, right? But how about if I lose an arm in an accident? There will not be a new arm growing out. So we need to help nature with new organs. You see a lot about these type of things in the literature. And trust me, I'm a strong believer of bioprinting. But sometimes I think we are talking about the future. So can we do like this cartoon is illustrating? Can we bioprint an entire heart today? No, we cannot do that. I think we will be able to do beautiful things like that in the future. But I'm sure it will take a few decades before we do a full heart with a bioprinter. But what can we do? We cannot create new organs maybe, but we can personalize organs and make them clinically useful. And that's what I'm going to tell you about now. There is always a problem we have to solve. And the problem I'm trying to solve is about transplantation. And as you know, we are transplanting hearts and livers and kidneys every day all around the world. However, there is always a big cost coming with transplantation, and that is about the immune system. In order to transplant without our bodies kicking out the new organ, we have to suppress the immune system for the rest of our lives. And that is quite efficiently done, but again, there is a high cost. Living without the immune system is in itself costly. We cannot be in big crowds. We can be very sick by a simple flu. And the drugs that we are using are connected to the most severe side effects we know about drugs. For example, dramatically increased risk of cancer. So how about risk balance? Many life-threatening organs we tend to transplant, but a lot of other organs we don't transplant because of this risk-benefit balance. So how can we do? We can do like this. We can personalize the transplant. And this is how we do it. If you start on the top left with the blue guy, he's the donor. He's donating something. In this example, it's a blood vessel because that's what we are doing now. And we are doing a classical decellularization. We take off all the cells and DNA to get off the identity of this transplant. What is left when you take away all the cells? There is a scaffold. There is always something when you take off the cells. And interestingly, most organs look more or less identical when you take off all the cells. This scaffold is then the perfect building block for the next process. And our next process starts with a red guy. And here is where we are unique. We are doing something that we call a personalization process. We are treating this scaffold coming from the upper line with blood from the patient. And that's where we are unique. We are using peripheral blood from the patient. We are not isolating out any cells. We are not manipulating the blood in any other way, that we are just adding some enhancing factors. So blood with some enhancing factors are key to our personalization process. And in contrast to people that are using pure population of cells that you have to isolate, identify, expand, things that takes a long time, is very costly, and regulatory-wise, it's a nightmare. Our process of personalization is only 10 days long. Very low costs, and the regulators are happy because we are not manipulating any cells on our way. So the personalization are ending up with something that we can transplant without waking up the immune system at the patient. So how about what we are doing then? This is exactly the whole idea with this process. So we are starting our clinical journey here. In the groin region, we are having arteries and veins, and the arteries are leading blood down to the feet, and the veins are leading blood back to the heart again. Interestingly, we are animals on two legs, so the blood are constantly trying to, by gravity, go down to the feet again. And this is a problem. And we have solved that by having one or several pair of valves in this groin region. And as you can see, these valves are simple flaps that are opening when the blood is pumped up and are automatically closing when the blood is trying to go down again. But what if these valves are failing? And believe me, more than 6 million patients in only the US and Europe are having failing valves, and a disease called chronic venous insufficiency. And the symptoms starting with swelling of the legs, pain in the legs, but gradually and unfortunately, open bleeding leg ulcers are formed. And they can look as terrible as this. And you can imagine this is a disease that are causing a lot of problem for the patients. They are typically on sick leave, early retirement, quality of life is really bad. So what is our solution? This is our solution. A fully functional piece of this vein with functional valves coming from a donor. But again, we will not transplant if we have to live without an immune system for the rest of our lives. But our patient can receive this graft without use of any immunosuppressive agents. So this is a little bit from reality how such graft looks like. So this is our product. We can have this graft going in our lab for many, many days. But we have also now started our clinical trial side of the business. So a little bit more than a year ago, we started our first-in-man trial in Spain. It's a small trial. It's a phase 1-2 trial, so it's 15 patients. And as you can see, we have now transplanted a little bit more than half of the patients in this trial. And now to the good news. So this is a typical example of a person or a patient with chronic venous insufficiency. You see a nasty leg ulcer that is not healing. And the upper illustration is before surgery, and down it's only 4 weeks after surgery. And next one, as you can see, 3 months later, the ulcers are completely healed. An even worse example is this one. As you can see, these leg ulcers can be really, really terrible. And you see the short time windows here. Second photo is 2 weeks, and the third photo is 3 weeks. So you see how rapid these ulcers are being healed when we have a fully functional valve in place again. So these are early days, but as you can see, data are really encouraging, and we are so happy that our patients are quickly recovering from a severe disease. So just the statements from our lead PI. So now to something very important and interesting. As you could hear, we are basing our product on donated human material. But the question I often receive is, can you supply a bigger market with human donated material? And the honest answer is we don't know, but we have a solution, and that solution is in 2 steps, I think. So what we have in late preclinical testing now is xenogenic material, so food grade animal tissues. So our second product will be an artery, and arteries are more or less identical between cows, pigs, and us humans. So we can easily use a bovine artery covered with the patient's material in our personalization product. So then you can understand that our product model is much easier if we can use the same animals as we use for food production today. But in the future, we think there is yet another step, and as I said, I'm a strong believer in bioprinting, and we have started to bioprint or 3D print these scaffolds. So these scaffolds are consisting of proteins, so they are relatively simple. So we have now in our labs the first arteries that are bioprinting that will be covered with the patient's own material in our personalization process. So our arteries program is, of course, a very important program because it's targeting a huge medical need. We are also doing other things with our product pipeline and our technology. For example, we can regenerate the peripheral nerves, heart valves, and other types of tissues. So this is really a platform. The business model is very simple. Verigraft will produce personalized tissue-engineered grafts and commercialize them in Europe and North America. And of course, key to this, I'm coming back and emphasize that once more, hurdle in this area is very often cost of manufacturing, putting the reimbursement model at risk. But our cost of manufacturing is very low, time of manufacturing is very short, and regulators are happy. So we see our reimbursement model to be very strong. Just before I end up, I would like to mention that we are located in Sweden. We have a lot of interesting programs ongoing in Sweden, so Sweden is a good place to be in at the moment. The government are supporting us in many ways, and bottom left you see something important. I think we may have people here in the room from CCRM Nordic. That is a Nordic branch of CCRM in Toronto. So a lot of interesting things going on. In Sweden, we have also attracted a lot of grants over the years in our company. So coming to the end, as most small companies, we are more or less always looking for new funds. I think it's especially good timing now to invest in our company based on the very positive clinical data we are currently receiving. So as a repetition, there is an enormous medical need for transplantation tissues. There is an enormous market. Some of the market, I didn't mention that, the market for arteries is today occupied by big companies selling synthetic grafts. Gore, Medtronic, and other companies are selling synthetic grafts, working in some cases, but also with a lot of disadvantages. So a few other arguments for why you could invest in our company right now. So why not end up with a lobster again? Don't forget the lobster. The lobster can regenerate organs. We cannot, so we need help. So why not keep an eye on Verigraft? There you see, two big claws. So thank you very much for your attention.